Hello, thank you for joining the Health Effects Institute 2021 webinar series. My name is Joanna Keel and I'm a research assistant here at HEI. Welcome to our Early Career Spotlight, where we will hear from winners of the 2021 Jane Warren Trainee Conference Award. Before we delve into today's webinar, I'd like to make a few housekeeping announcements. All attendees are muted for the duration of the webinar. There will be a 6-minute Q&A period after each speaker, and you can submit questions using the Q&A function at any time during the webinar. You can also upvote questions in the Q&A. If you experience any logistical difficulties, please contact us using the chat box. And to help us improve our webinars, please complete the post-webinar feedback survey, which will be emailed to you tomorrow. It is my pleasure to introduce to you our chairs for today's session, our Deputy Director of Science, Annemoon van Erp, and our new Director of Science, Ellen Mantis. I will now pass it over to Annemoon to tell you a little more about Ellen, as well as Jane Warren, the person in honor of whom we have named this award.
There we go. Thank you, Joanna. Welcome, everyone. First, I'd like to say a few words about Jane Warren. Jane came to HEI during the early 1980s when HEI's research program was just getting underway. She was instrumental in building its research program on air pollution and health, including a landmark multi-center clinical study on the health effects of carbon monoxide. She was Director of Science at HEI from 1999 until her retirement in 2008. I had the pleasure of working for her during that time. She was an inspiring leader who led by example, an incredibly hard worker and excellent communicator. Jane thought it was very important to draw young people into science and public health, and was a driving force behind the creation of our Walter Rosenblith New Investigator Award, a program that has by now awarded almost 30 early career scientists. Unfortunately, Jane passed away before her time earlier this year. By naming the HEI Travel Award after her, we hope to continue her legacy to provide early career support for many years to come. I would like to congratulate the 5 presenters today and look forward to hearing about their research. Now let me introduce Dr. Ellen Mantis, our new Director of Science, who will co-chair the session with me today. We're very pleased that she has joined HEI in this role. She started last week, actually, and look forward to working with her very much. Before we get started, I would like to thank Joanna Keel, Pallavi Pant, and Eliane van Vliet for organizing today's webinar. With that, I'll hand the mic back to Joanna to start off the webinar.
Thanks, Annemoon. So the Jane Warren Trainee Conference Award has evolved from our annual Student and Postdoc Travel Award, which has been given to early career researchers at our annual conference for the last 4 years. In non-pandemic times, we would fly the awardees out to the conference and they would present posters on one of the 3 days of the event. In lieu of that, the awardees this year will receive a $500 cash prize to be used for professional development activities, such as membership in a professional society, attending a workshop or conference, or undertaking professional development courses. And of course, we also get to hear from them about their research today. We had 52 applicants this year from a range of institutions and countries, and without further ado, I'm happy to announce the winners of the Jane Warren Trainee Conference Award 2021. Ekta Chaudhary from the India Institute of Technology in Delhi, India, Daniel Mork from Colorado State University, David Luglio of New York University Langone Health, Dr. Gage Kerr of George Washington University, and Weran Nushi of University of British Columbia, Canada. We'll now pass it back over to Annemoon to introduce the first of our speakers. Thanks, Annemoon.
Thanks, Joanna. All right. I'm very pleased to introduce our first speaker today, Ekta Chaudhary, a full-time PhD scholar, Honor Professor Sagnik Day in the Center for Atmospheric Sciences, IIT Delhi, India, where she studies the effects of air pollution on health. Ekta has been an integral part of the Indo-UK project Daphne, Delhi Air Pollution, Health and Effects, since 2018. She is currently associated with Earthwatch Institute India Trust, EIIT Delhi, as a research expert on air quality and human health, where she reviews and develops specialized courses, training, and workshops for students, teachers, and residents. As a budding enthusiastic researcher, Ekta looks forward to exploring and identifying intrinsic correlations between various health impacts and air pollution by seeking extensive collaborations with biomedical scientists. The title of her presentation is Association of Ambient PM2.5 Exposure and its Composition with Anemia Prevalence amongst the women of reproductive age in India. Ekta, thank you for joining us all the way from India today. We look forward to hearing about your research. The floor is yours.
Thank you so much. Let me share my screen. Is it visible to all of you?
Yes. Thank you.
Great. Hello, everyone. Thank you for having me here. It's a pleasure and honor to get this opportunity and presenting my research work among esteemed researchers of HEI. I would like to thank the HEI committee for this award and would also like to congratulate my fellow awardees. On that note, let's begin. My research is about the association of ambient PM2.5 exposure and its composition with anemia prevalence amongst the women of reproductive age in India. Let's do the situation analysis. Oh, it's not moving. Wait a second. It's not moving.
I can see your first slide.
Yeah, but
it's not working.
I can share your slides for you.
Just a second.
Joanna, you want to share and forward them?
Yes, Ekta, do you want to try one more time?
Yeah, okay. Let me see.
I'll share your slides.
Great.
Okay.
So let's do the situation analysis of air pollution and health effects scenario in India. Air pollution is a critical threat to human health in India. It has been a major health risk with ambient air pollution and household air pollution. There are many studies focusing on concentration, composition, and health effects of air pollution, but most of them are focusing on personal exposure. There are studies which are focusing on mainly on coarser particulate matter, PM10, and only few studies focusing on PM2.5. If you look into the literature, we have most of the studies covering time series analysis. If you look at the right side, the India map, only handful mega cities are covered in detail for health effects of air pollution. So yes, we do have priority gaps and we are lacking with geographical coverage of health effects of air pollution studies, and we need to make some strategies to facilitate the clean air actions. Next, please. Yeah, there are health effects of air pollution. Short-term effects include respiratory infection, reduced lung function, allergies, bronchitis, skin irritation, and all of them. Whereas long-term effects include COPD, lung cancer, cardiovascular diseases, preterm delivery, diabetes, but we don't have any pan-India scale coverage for anemia. And although our group has already established a study where they have shown the association of ambient PM2.5 with anemia prevalence under age 5 for children. Next, please. Yeah. So anemia is a condition where RBCs are lacking in number or dysfunctional. So that leads to reduced oxygen level in body organs. Anemia can be of temporary or long-term. It can range from mild to severe depending on the hemoglobin level. The motivation behind my study was the anemia prevalence in India. Next. So I found that anemia prevalence in India was 53%. And if you see the first plot where we have did the anemia prevalence by wealth index. So the poorest people population are having more anemia prevalence than the richest one. Next. And then we subdivided this anemia prevalence to their categories, where it is divided into mild, moderate, and severe on the basis of hemoglobin level. Next. So we found that 41% of WRA, women of reproductive age, are having mild anemia, 12% moderate anemia, and 1% severe anemia. In this analysis, till now, we have only considered anemia prevalence, but not associated PM 2.5 here. Let's see what happens when we add exposure values or PM 2.5 exposure to our analysis. Next. So the methods and data sets used, we have health data set. I have used two Indian national level data set. First is NFHS 4. So in which anemia in WRA and their socio-demographic factors were obtained. We have a series of National Family Health Survey. Out of those series, I have taken NFHS 4, which is available at district level, just to add nutritional impact or add nutritional parameter to my model. I have used another health data set, which is National Sampling Survey of 68 version. Next, please. So yeah, in the second health data set, we have taken dietary iron intake as a proxy of diverse dietary score. If I talk about the exposure data set, I have taken 1 by 1 kilometer resolution PM 2.5 exposure. Our group has already established this data set. For more details, you can look into Day et al. 2020. Next. Our data includes population of around 7 lakhs subjects and of age 15 to 49 years. Our data, next. Our data includes basically logistic regression adjusted for potential risk factors like wealth index, education, BMI, cooking fuel, and all of these covariates. The analysis is basically divided into two parts, ecological analysis and individual level analysis. Whereas in ecological analysis, we have looked into the association of anemia prevalence and ambient PM 2.5 at district level only. During the individual analysis, we have taken geocodes for the population, which was available for around 28,000 subjects. And we extracted the PM 2.5 exposure for those 28,000 geocodes and then clustered by using primary sampling units. Next. Yeah, so impact of chronic ambient PM 2.5 exposure on anemia prevalence. Left plot shows the anemia prevalence at state level. So you will see more than 60% of states like more than 60% anemia prevalence in states like Jharkhand, Haryana, West Bengal, Bihar, and Andhra Pradesh, whereas very less than 1/3 in Mizoram, Manipal. So the right side map is for anemia prevalence and PM 2.5 exposure at district level. We have spatially plotted this. PM 2.5 exposure in our study is decadal, starting from 2007 to 2016. Yeah, next please. Looking into the exposure response curve, we have log odds of anemia and decadal ambient PM 2.5. We were looking into the relationship by using this line. You can see the detrimental effect starts from 30 to 35 microgram per meter cube. Next. This also established a relationship that for every 10 microgram per meter cube increase in ambient PM 2.5 exposure, we have anemia prevalence of around 7.23% increase. So we have an increase of 7.23%, which is a big percentage. And recently our group has already established this relationship for child of under age 5, and they found out that for every 10 microgram per meter cube increase in ambient PM 2.5, the children of age under 5 are having 1.9% of anemia prevalence. So we looked into the hemoglobin and ambient PM 2.5 relationship also. Next. So this says there is a significant decline when we increase the ambient PM 2.5, the hemoglobin levels decreases. Next. Yeah. So at individual level, when we looked into hemoglobin and ambient PM 2.5 exposure, here also the decadal PM 2.5 exposure is used. So at individual level, for every 10 microgram per meter cube increase in ambient PM 2.5, we have a 0.044 gram per deciliter decrease in hemoglobin. So by this exposure response curve, we are saying that by increasing the ambient PM 2.5, we have a certain percent increase in anemia prevalence, and therefore we have a decrease in hemoglobin also. Next. So we looked into the effect modification. Effect modification is the situation where the magnitude of the effect of an exposure variable, which is PM 2.5 in our case, on an outcome variable, which is anemia prevalence, differs depending on the third variable. The third variables are all these covariates or parameters we have looked into. So starting from the bottom, we have wealth index, which is divided into poorest, poorer, till richest. So if you remember, I have shown the relationship of anemia prevalence with wealth index. They showed that the poorest population were having more anemia prevalence, but there we didn't include ambient PM 2.5. But while we add the ambient PM 2.5 into our analysis, the result comes exactly opposite. So, and then urban population were having more anemia prevalence when exposed to chronic ambient PM 2.5. So we don't have exact literature findings which make our analysis, this particular effect modification analysis, stronger. I would also welcome comments from your side, which can enhance my analysis for this particular map. Next.
You have less than a minute left, Ekta. Can you please wrap up?
Yeah, sure. Yeah. Looking into PM 2.5 composition, we have used MERA2 reanalysis dataset, which was available at 0.5 to 0.6 degree. But as we used 1 by 1 kilometer satellite dataset, I have regraded MERA2 reanalysis dataset to 1 kilometer. And we looked into interquartile range for each of these components. And what we found out that at each at ecological level, for each IQR of BC, we have anemia prevalence increased by 27%. And then for sulfate, it was 28%, dust 20%, and then OC organic carbon, it was 13%. Similarly, so yeah, I would like to add that this MERA2 reanalysis data was also validated with ground-based measurement. We have a paucity of ground-based measurement, but we could only find one in situ site where we did this validation, and we have a significant R square value also. Next, please. So wrapping up here, we established a chronic relationship between ambient PM 2.5 and anemia prevalence, which also showed that there is a higher impact of BC, sulfate, and all these components. We recommend that the cohort studies should be implemented to understand the biological mechanisms and toxicity issues. We are also doing some analysis, extending this current analysis by using policies like NCAP and PMUY, that is Pradhan Mantri Ujjwala Yojana, to make this analysis more efficient and to reach the target of anemia free, which is a 35% anemia prevalence. So earlier, now it is 53%. We want it to reach 35% to reach this anemia free target. Thank you all. That's all from my side.
Thank you very much, Ekta, for this very interesting presentation. let's go to the Q&A here. The first question is from Alison Patton, who writes, "Hi, Ekta, thank you for this great presentation. Besides air pollution, what are other major factors that influence anemia prevalence?"
Yes. So anemia prevalence, in the literature also we have looked that anemia prevalence is enhanced by less or deficiency in iron intake, and we have taken that parameter also, and there are more health issues which can lead to this anemia prevalence. Also, patients with CVDs also are more prone to anemia.
Sorry, could you say again, patients with what condition?
Cardiovascular diseases.
Cardiovascular, okay, thank you, yes. And are there, just to follow up, are there ways that these iron deficiencies are being addressed?
Yes, in India, there are policies running up which are looking into iron fortification. So they are providing iron supplements and other biological medicine kind of supplements to enhance the iron level in women or the people who are anemia prevalent. So there are policies going on in India.
Thank you very much.
To tackle this issue.
The next question is from Karan Vora. Great presentation, Ekta. Are any specific reason why the log odds of anemia are so low for ambient PM 2.5, around 30 to 40 micrograms per cubic meter?
Yes, while doing our study for child anemia also, we were used the same methodology, and I cannot say why it is coming very low log odds, but we were seeing the same result. So 20 microgram per meter cube PM 2.5 exposure was coming for children, and then 30 microgram per meter cube exposure is coming for women. So I don't have exact statement for that, but yeah, we are seeing the similar relationship with other studies also.
Thank you, Ekta. And I'll combine the next questions, which is about the prevalence of anemia in the population. Is 60% considered high? And what is the difference between men and women here?
So yes, we were planning to do this analysis among adults, which includes both men and women. We haven't add men data here, but we know that anemia prevalence in men are around 48%. Okay. I'm not sure about the percentage, but we divide this anemia prevalence on the basis of hemoglobin level. So if I say any anemia, so that in case of women, then hemoglobin level should be below 12 gram per deciliter. For men, it could be starting from 13 gram per deciliter.
Okay, that's very helpful. Thank you very much, Ekta. I think we're coming to the end of your Q&A session. Thanks again for joining us from India. And I'll hand off the microphone to Ellen to introduce the next speaker. Thank you.
Good morning, everyone. It's a pleasure to be here today. I have the pleasure of introducing Daniel Mork. Daniel is a PhD candidate at Colorado State University. He researches maternal environmental exposure effects on prenatal development with advisor Ander Wilson. Following graduation this summer, Daniel will begin a postdoctoral fellowship at Harvard School of Public Health, where his research will focus on understanding the relationship between air pollution and Alzheimer's disease and related dementia. He'll be talking today on estimating perinatal critical windows to environmental mixtures via structured Bayesian regression tree pairs. Daniel, the floor is yours.
Thanks for that introduction, Ellen. I'm very excited to be with you all today. I'd like to thank the Health Effects Institute for selecting me for this Jane Warren Award. It's an honor, and I'm very happy to be able to share this work with a lot of esteemed researchers today. So my work today on estimating perinatal critical windows to environmental mixtures is primarily methodological focus. So we've developed some methods to do this. But throughout this presentation, I'll present both our methods as well as the data analysis we did using our methods. So to begin, I'd like to just describe perinatal critical windows and then some of the data that we're going to be looking at. So we know that there's strong evidence that an expectant mother's exposure to a variety of different chemicals during pregnancy is associated with changes in birth and children's health outcomes. So our data, as seen here, consists of weekly exposure data for 4 different pollutants and temperature, which is taken at the mother's census tract of residence. And we're interested in how changes in these different exposures are related to measured birth weight, in this case, in terms of our data analysis. But this could be used for any birth or health outcome, as well as a lot of other scenarios. So a majority of research on perinatal exposures for mixtures often uses trimester or pregnancy average exposure measurements, which we know can lead to some bias in the estimates. And it also precludes answering a bigger question of precisely when during pregnancy are exposures related to the outcome. So answering this question can help us to understand these developmental critical windows when changes in exposure can alter those health outcomes. Furthermore, this can give us insights into particular biological mechanisms that might be altered by those chemicals or other exposures. There's further nuance that exists in these exposure effects if we start to consider interactions between exposures. And these interactions could take place either at the same time period or across different time periods. Early exposure to one pollutant, late exposure to another pollutant, where we might have some sort of priming effect, where the early exposure alters a phenotype that increases susceptibility to a later exposure. So analysis for a single pollutant is often performed with a well-known distributed leg model, which can take and find those time periods during exposure when those changes in exposure alter the outcome. But there's no methods that exist for 3 or more pollutants. So our methodology development centers around these 3 primary goals. So first of all, to estimate the exposure effects, interactions, and critical windows due to all of these mixtures of exposures, to identify the harmful components, those components that are most related to the changes in the birth or health outcome. And then third, to control for these confounding co-exposures. We know that there's a lot of different things that could go on kind of relating all these co-exposures. And so we want to be able to control for that entire time period of exposure for the other exposures while understanding the effects of one particular exposure. So next, I'm going to just give you an overview of our methodology that we've developed for, first of all, a single exposure distributed leg model, and then looking at this idea of mixtures within a distributed leg model. So to begin, let's look at our single exposure method. So distributed leg models are often estimated using spline methods or often Gaussian process methods. And so we've developed a new technique for estimating a distributed leg model using a regression tree. So in this method, a regression tree partitions the time span of exposure into these sort of discrete segments where the number of change points and then the location of these change points are all learned by the model based on the data. Having few change points along this time span introduces a necessary constraint in the distributed leg model to deal with temporal correlation in the exposure data. And then also, within each of these time segments that the model finds, we have a piecewise constant effect. So this method has several advantages over, for example, spline-based methods, where we're able to eliminate the necessary smoothness constraint, where in the case of maybe like a hard critical window, where there's kind of a very well-defined start and stop time of that critical window, we're able to estimate those much better than the spline-based model. The spline-based model or other smoothing methods tend to over-smooth and overgeneralize these critical windows, placing them where they don't actually exist. To allow for smoothness when necessary, we use an ensemble of these regression trees that are each able to break up the time span slightly differently and to accommodate smooth effects. And we found that in simulation, our tree-based distributed leg method actually outperforms the established spline and Gaussian process distributed leg methods. To extend this method to mixtures, what we do is develop a concept of tree pairs. And so in these tree pairs, we have two different tree to distributed leg models that estimate two separate main effects for two different exposures. Now, as before, the time breaks and the number of time breaks and their locations are learned by the model. But here, the exposures that get estimated by these tree distributed leg models are also learned by the model according to the data. After we have these two main effects, the structure of these trees define an interaction surface across time. And so with respect to this interaction surface, we can kind of think about the x-axis here as being the time of exposure 1, the y-axis as being time of exposure 2, and then we have this piecewise constant effect across the surface where we were doing kind of a lot of dimension reduction here. Instead of estimating these t-squared different parameters, we're kind of breaking this down into a simpler portion. So here we're just estimating 12 different effects across time. Furthermore, by allowing different exposures to be included in the different trees, we introduce a variable selection where if a exposure isn't included in a tree, then it gets selected out of the model. And this occurs through an ensemble of tree pairs. We have lots of different tree pairs in the model, and they're estimating different main effects and different interactions based on what's in the tree. And by removing an exposure from a tree entirely, we select exposures out of the model. And then finally, we introduce some exposure and interaction specific shrinkage priors on the model where we can have a main effect, say, but no interaction effect, or an interaction but no main effect, while still maintaining sort of an interaction hierarchy where whenever an interaction is included, those main effects are also included. Okay, turning back to our data analysis now. So in the data analysis, we looked at just about 200,000 full-term births in the Denver, Colorado metro area. And we looked at birth weight z-score. So it's a birth weight standardized for sex and gestational age. And for each individual birth, we had 5 exposures measured weekly at the mother's census tract of residence. And we controlled for a variety of different things located from the birth certificates that we had data of, demographic factors, some spatial factors, as well as temporal considerations. Okay, before looking at the actual effects we found, I want to just kind of look at some variable selection. And so of these 5 different exposures, our model selected 4 as being kind of highly correlated with the outcome. So temperature, carbon monoxide, sulfur dioxide, and fine particulate matter. And so this plot here I'm just showing you gives us 2 different measures. It gives us a posterior inclusion, which is the probability that the exposure gets used in at least one of these regression tree pairs. And then on the y-axis, we have the effect size. So we can kind of judge the effect size of these different exposures relative to each other. So those were the 4 main effects, but we also note that we detect an interaction between fine particulate matter and temperature. And so we'll take a look at that as well.
And you have about a minute left if you want to wrap up.
Thanks, Joanna. Okay, so just to take a quick look at our model that we're actually fitting. So we're fitting this additive distributed leg model, which I'm not going to spend too much time on, and then all the possible interactions between any 2 exposures at any 2 times. And so we detect critical windows for 3 of these different exposures: fine particulate matter, sulfur dioxide, and temperature, as well as this interaction effect, which I'm going to spend a little bit more time on. So if we think about the time period of exposure to fine particulate matter and the time period of exposure to temperature, we detect interactions both at the same time for both, but then also at different times. So earlier exposure to fine particulate matter and later exposure to temperature. And this is maybe hinting at sort of a priming effect where the early exposure to fine particulate matter is increasing susceptibility to temperature later on. Okay, so just to summarize our model a little bit, this methodology development allows us to do a bunch of different things within this mixture setting for distributed leg models. First of all, we're able to do component selection, take into account pairwise interactions and quadratic main effects of possible nonlinearity. It's easy to interpret because of the structure of the model. It's a very interpretable machine learning approach. we can deal with a much higher dimension of different predictors we're measuring than the number of observations if needed, and consider continuous and binary outcomes. In a simulation that we have in our paper, we showed that we have high precision in critical window identification, and we're able to outperform existing distributed lag methods for a single exposure while still scaling well to mixtures. Some limitations here is that we're only able to account for two-way interactions, so we can't have these highly complex exposure responses. And just to give you some computational details, our method is based on the Bayesian additive regression trees method, but it scales very efficiently in all the different factors that we might be interested in: sample size, number of exposures, and the time periods of measurement. So whether you're fitting for 1,000 observations or 100,000 observations, this model can be fit in a matter of minutes to hours. Just to give you a sense of related work, we have some other work out there using these distributed lag models for measuring nonlinear effects as well as heterogeneity in effects. And the latter one will be coming out shortly. So thank you all for your time. I'd like to just give a shout out to my advisor and then some funding grants for this work.
Thank you, Daniel. We have a number of questions. That was a great presentation. Let me start with some of the questions here. The first one comes from David Savitz. He says, this approach seems elegant, but there's really not an anchor of certainty on whether it's providing valid results for these exposures and outcomes. Are there any opportunities to use it with either positive controls or negative controls, detecting patterns that could be valid, such as exposures in the wrong place or wrong time?
Yeah, so that's a good question, because it does kind of get at, we want to make sure that we're detecting what we want to detect here. And so the part of the work that I didn't have time to present to you today was a bunch of different simulation analyses that we did do, looking into some of these questions, whether, if we have multiple different exposures, are we detecting the right exposures? If we have different critical windows, are we detecting the correct critical windows? And what we found was that our model does a really good job, first of all, in detecting critical windows precisely. And so if there's a critical window detected by the model in our simulation, there's a very high probability that that critical window is a true critical window. And this outperformed a lot of competing methods in the single exposure setting, but then it scaled into the mixtures as well. With regard to mixtures, some of our simulations looked at both like main effects and interaction effects, and whether we were detecting the correct exposures, the correct components in the model. And what we found was that, yes, the model is detecting those with that kind of response of the very high posterior inclusion probability relative to the other exposures in the model, which were included into the model at a much lower probability. And I would definitely encourage you to kind of look at our paper for some more of those simulation results. We looked at some kind of more extensive ones as well, looking at kind of more complex interactions between multiple things that aren't included in the paper, and it seemed to be fairly robust in those settings as well.
Thanks, Daniel. The next question comes from Jana Milford. Great presentation and very interesting method development. Could you say more about the pollutant exposure measurements you used?
Yeah, of course. So the pollutant exposure measurements came from two different areas. The first one was the fine particulate matter, which was came from US EPA CMAQ models. And so those, I believe, were put at the centroid of the census tract. And then the rest of the pollutant measures, we used all available EPA monitors in the area, averaged by week. And then we used inverse distance weighting to put those back at the mother census tract of residents.
Thank you. Next question is from Mode Ibrahim. It says, hi, Daniel, thanks for your great presentation. Any reason you did not include ozone in your analysis?
Yeah, so this is a good question. Actually, early on, we were looking at ozone as well. But one of the issues with ozone we found was that it was so highly correlated with temperature that the temperature and ozone sort of had like competing effects where we didn't trust the results when including both of those. So we kind of said, okay, we're going to include one or the other in this particular analysis. And because this was a methodological paper, we didn't spend a lot of time kind of exploring that question, but it is definitely a question that I think should be given more attention.
Thanks. And I think we have time for one quick last question. Yang York says, great presentation, Daniel. Was the exposure timeline 36 weeks of gestation?
We used 37 weeks of gestation. So we did do a stratified analysis where we only included births that exceeded 37 weeks or more. And so then we included the entire 37 weeks of gestation, or of exposure data. And so if a birth did occur at 40 weeks, we only had their first 37 weeks of exposure data as well. So everyone had the same amount of exposure data.
Okay, thanks so much, Daniel. I think we've run out of time, and I'll turn it back to Anna Moon.
Thank you, Ellen, and thank you, Daniel. Our next speaker is David Luglio, who is a PhD student at New York University Langone Health, studying environmental health. He earned a Bachelor's of Science in Biological Sciences from Cornell University. His research focuses on air pollution and cardiopulmonary health. He works on a range of projects, and currently the bulk of his work concerns the air quality in subway systems, but he's also conducting a coronavirus study. The title of his presentation is PM 2.5 concentration and composition in subway systems in the northeastern United States. David, please go ahead.
All right, thank you. So today I'll be presenting my initial research into the air pollution, namely PM, of subway systems in northeastern United States. But first, I'd like to just reiterate that exposure to PM has health consequences. It is well known that chronic exposure leads to cardiopulmonary morbidities and mortalities, but we also see physiological changes from short-term high exposures that can range from blood pressure changes to asthma attacks and myocardial infarctions in sensitive individuals. And that's one of the reasons why we have both 24-hour and annual PM standards set by the EPA. However, the toxicity and mechanisms of action of the PM is expected to depend on its composition. A metal-rich particle is going to have much different toxicity than a soil particle. Subways. So subways are great. They're great for cities. They can move a lot of people at a time in a fast, cost-efficient manner, and they keep cars off the road, which reduces not only congestion, but street-side air pollution levels. But they are dirty, and many of us know they are dirty. And this is not limited to visible grime, rats, and garbage, but includes the air. Studies around the world have shown that PM levels on underground platforms are many times ambient levels and are metal-rich. But unfortunately, human health studies are few and far between. In particular, there's actually been very little research in subways in the northeastern United States, both in terms of measuring concentrations and composition, as well as doing any health studies. And given the volume of people affected, I thought it'd be a good idea to investigate these systems. My research focuses on the subways of New York City, including PATH, which connects Manhattan to New Jersey, the MTA, or the stations run by the City Transit Authority, as well as subways of Washington and Boston. So methods. Well, today I only be presenting the PM 2.5 concentration and composition data with some mentions of future studies for health. So our team and I visited over 70 subway platforms spread across the multiple systems and recorded PM 2.5 concentrations for 5 to 10 minutes at a time with a photometric instrument, and each platform was sampled 4 times each. This photometric data was corrected with gravimetric fetal samples later on. At 2 stations with the highest PM levels identified in Boston, Washington, and the PATH system, we collected filters. These filters were analyzed by trace element composition by X-ray fluorescence, and for carbonation compounds by OC-EC analysis. And a handful of these filters were analyzed further with a synchrotron, which gives us more information about the oxidation state and mineralogy of each individual element, and really of individual particles on the filter. So this is a box plot showing the PM 2.5 concentrations at only the underground stations. The y-axis is the PM 2.5 concentration in micrograms per cubic meter. I also have listed, sorry about that. Also have listed here the Long Island Railroad, which connects New York City to towns on Long Island and the Philadelphia subway system, even though my research doesn't really focus on these two systems. And what is clear here is that the PM levels exceed the average ambient concentration measured in each city by a lot. And we measured just about the chute side levels, average again through all cities to be about 15 micrograms per cubic meter. But the amount it exceeds is quite variable. You have the path system, where you have very high concentrations. In fact, we had one sample which is almost as high as 1,500 micrograms per cubic meter. And you have some stations where some systems it's actually very low, as in Philadelphia. The mean concentrations for each are 327 for Boston, 341 for Washington, 548 for the MTA, 779 for PATH, and about 112 for Philadelphia. New York City seems to have the worst subways. Boston and Washington seem to have very similar ranges. So this pie chart shows the composition of the PM2.5 average between all filters. The wedges shown represent contribution to total PM 2.5 mass. And what is obvious is that the material is dominated by iron, about 52%, although this did in fact range from about 40 to 70% based on location. TC stands for total carbon, and represents a little under a third of total mass, although this also did vary between different stations. So there are a bunch of elements where we do have at least 1%, they contribute to 1% of total mass, and that includes barium, sodium, magnesium, sulfur, silicon, and calcium. Metals such as manganese, chromium, copper, and nickel were also present at very small but detectable amounts. So composition is important to identify the potential toxicity or mechanism toxicity of the PM, or to actually determine the sources of the air pollution. But simply stating the presence of a certain element is really not enough. We have to recognize that there are many different forms of element which have different effects and can come from different sources. For example, chromium-3 has much different toxicity than chromium-6. And that's why, that's why we did a deeper analysis. So presented here is the mineralogical composition of iron-containing particles. At the bottom, we have listed stations. So Nicholas Street, Newport from PATH, White Wayne Government Center from Boston, Van Ness and Capitol South from Washington, and I also have the listed average. And what's striking at first is that each of these boroughs are very similar. Although total iron concentrations may differ between stations, the relative concentration of different forms of iron to total iron actually does not differ that much at all. So important things to point out, hematite and magnetite, which range anywhere from 30 to 40% of the total iron, are generated in high energy environments, which are somewhat reminiscent of the conditions caused by grinding of train wheels against their rails, or the brakes against their wheels, or collector shoes in contact with an electrified third rail. Ferrihydrite and goethite are generated in cold environments and are indicative of rust. It seems about half the iron seems to have been sitting on platforms rusting, which does take a lot of time, which is an indicator that the stations have not been cleaned. And finally, only a small amount of this iron is found as Fe2+, which is found in magnetite and wacite. Fe3+ and Fe2+ may not have different intrinsic toxicity, but Fe2+ is much more soluble, which poses a much larger biological threat. So from concentration data, I can estimate the increase in health risk. And here I only looked at cardiovascular mortality. The subway riders compared to people who do not use the subway, but still live in the same area. And here I'm only considering the PATH system. So the average underground concentration in PATH platforms is 779 micrograms per cubic meter. And let's say a commuter regularly sits on the underground platform for 15 minutes a day, and an employee works at least 2 hours per day underground. The rest of the day, we consider they are exposed to the average ambient concentration in New York City in 2019, which is 7.7 micrograms per cubic meter. Run simple time average calculation shows that a commuter and workers exposure is increased to 15.7 and 72.0 micrograms per cubic meter, respectively, from a base concentration of 7.7 micrograms per cubic meter. That is if they didn't use the subway at all. Pope et al. Estimates that there's a 6% increase in relative risk for cardiovascular mortality with every 10 microgram per cubic meter increase in PM. Using those numbers, I can estimate there's an increase about 4.8% risk for commuters and 38.6 increase in risk for workers from cardiovascular mortality who are exposed daily to PATH PM. And this is overly simplified. It ignores a lot of other exposure events, such high exposure events that you experience daily, like through cooking, sometimes through vacuuming, and in many cities, there's a lot of other high emission sources. But it is really just a simple, quick look at the potential consequences of not cleaning subways. Currently, I'm beginning to start in vitro toxicology studies of where respiratory cells have been exposed to the subway PM. And I plan to do cardiopulmonary studies with volunteers and possibly transit workers in the future. So the ultimate goal of this work is to provide evidence for or against the need to remediate the subway air quality in order to protect people's health. Results so far are alarming. These are very high concentrations that we usually don't see in other environments. It's just, it's sometimes, 1500 is higher than the worst days in like Beijing or in Delhi. But this work is also, I would also like to identify the sources as in a way to provide possible solutions to the problem, at least knowing where to target to protect, to reduce the air quality on the studies. And really, I would like to continue work like this, which is the provide evidence of how and why policy needs to change when it's necessary to protect people's health. I do have some information about my own background. I do have a very broad background. I study a lot of biology, but right now I'm primary exposure assessment guy. And really, I think my future work is going to be mainly based on exposure assessment. And finally, some of my future goals, I plan to earn my PhD. Well, I would like to earn my PhD. My dissertation will really build on this work. In the end, the workers are the biggest concern because they spend so much time on the grants than anyone else. And then just some career prospects. And I'm leaning towards academia, but anything can really happen. All right.
Thank you very much, David. That was a very interesting presentation. We have some good questions lined up for you. I'll start off with the first one from Daniel Croft. Is there a correlation between the PM2.5 concentrations and the distance between subway stops, if platforms or exits serve as the main ventilation for the subway? Well, for example, does PATH New York City have the greatest distance between stops, or is there other reason for worse ventilation, tunnel under the river?
Well, we didn't look at this directly, but we do have some ideas. Yes, we do believe that the distance between stations might actually have a major effect. The dirtiest station in the PATH system, Christopher Street, is the first station outside of the tunnel going underneath the Hudson River. And in that coming in, there's actually a lot of turns, which can actually trap a lot of particles. So as the train comes in, it might be pushing a lot more PM into the station than otherwise. It's harder to tell because we didn't really measure all the other distances in between every other station, tell that's really commonplace. But the other reasons why the PATH in New York City might be more dirty is well, first of all, they're some of the oldest systems in the United States. Boston is older, but a lot of it probably has to do with management, how often they're cleaned, how often they're renovated. But you know, and really it's early to tell. We're definitely be looking at it in the future.
Mm-hmm. Thank you. There's a question from Dan Kraus. Just curious, he's curious to know if you personally traveled to all these cities to deploy the samplers, and if it was difficult to get permission from the transportation authorities to deploy these samplers.
Well, as my advisor always says, don't ask for permission, ask for forgiveness. No, we did not contact any of the transit authorities about this, but I did personally travel to all the cities except for Philadelphia, and I did do sampling on each of the lines. I had a whole group of people that helped me because it was hard just doing this by myself, but yes.
Right, so then you never got into trouble when they noticed you taking measurements?
I got looks from people, but generally, no one approached me. Definitely no officials have approached me.
Okay.
We are in more contact with some transit authorities now after some of this work has been published.
Mm-hmm. A question from Eloise Marais. David, great presentation. How do your subway PM2.5 measurements compare to measurements from cities in other countries, such as London or Paris?
Okay, subways in the United States are generally more dirty than subways around the world. London is probably the second dirtiest. Well, outside of New York City, London in the grand seems to be the second dirtiest that we can see, I can find. But the PM levels are still elevated in any of these other cities compared to the ambient concentrations, whether it's in Canada or South Korea or Japan or China or India or anywhere in Europe or Australia and stuff like that. Which it's still a global concern, and there's millions of people that ride a subway a day globally, so we should really pay attention to that.
Okay, thank you. A question from Jana Milford. A very interesting work. Impressive that you were able to consider iron speciation as a way of considering sources. How do PM10 levels compare to PM2.5?
So we didn't measure PM10 extensively. It is obviously higher, but it's surprising in the stations, 60% of PM10 is probably PM2.5, and that's what much we've measured so far, which is much different than we see in ambient. We see a lot lower percentage of PM2.5 being part of PM10, which is, part of it's interesting because I don't think most people would expect iron particles to be this small, at least PM2.5, if it's being generated by grinding or something like that. I will be testing in vitro toxicology of coarse particles and see if there's any difference between that and PM2.5 in the future.
Thanks. So we'll take one last question from Joyce Zhang. Can you infer particle age when it was emitted from the iron oxide speciation data?
Well, not precisely, but the different forms of iron. So goethite, and ferrihydride, which are both indicative of rust, they obviously had to be, well, they had to be oxidated from particles already there. Goethite takes much longer to form. So we know those particles are probably the oldest particles in the station. But we can't really, are these particles 10 years old or 5 years old or something? We can't really tell that. But we can tell that the particles have been sitting there for a while, some of them.
Mm-hmm. All right, thank you very much, David, for this presentation. I'll hand the mic back to Ellen.
Thank you.
Thanks, Santa Moon. It's now my pleasure to introduce Gage Kerr. He is a postdoctoral scientist in the Milken Institute School of Public Health at George Washington University. He focuses on ambient air quality, environmental justice, and the impacts of weather on air pollution in his research. Before GW, Gage earned his PhD from Johns Hopkins University in 2020, and his thesis focused on understanding the meteorological drivers of ozone pollution. He also holds a Bachelor of Science in Meteorology and a Master's in Earth and Planetary Sciences from Cornell University and Johns Hopkins, respectively. And his talk today is COVID-19 Pandemic Reveals Persistent Disparities in Nitrogen Dioxide Pollution. Gage, the floor is yours.
Thank you. And I assume that since you read my title, everyone's able to see my slides. So thank you all for tuning in, and thanks to the organizers for putting not only this session together, but for all the different sessions we've heard over the last few weeks. I want to give a shout out to the co-authors of this research, Dan Goldberg and Susan Annenberg, both at GW, as well as NASA for funding our research. I also, before diving in, want to get a couple definitions out of the way. I'm going to be talking about nitrogen dioxide pollution, and I'll mention NO2 in lieu of nitrogen dioxide. NO2 is generally associated with traffic and industry in urban areas, and it has many health impacts. I'll also be talking about NOx emissions, and NOx in this case refers to the chemical family that's comprised of NO and NO2, and it's very closely related to ambient NO2 concentrations. So to start out our talk today, I want to talk about trends in urban activities during the pandemic. The plot that you see here shows different sources or different activities that are very relevant for urban NOx emissions and NO2. And instead of just showing their trends during the pandemic, we have scaled each of these different sources by their overall contribution to urban NOx emissions to get both a sense for how they changed during the pandemic, as well as their overall importance for urban NOx emissions. So what I at least see when I look at this picture, the most stark thing I see is the light duty vehicle curve, which is shown in the dark green. Indeed, there was about a 50% decrease in light duty vehicle activity during the pandemic. And this sector represents an activity that's very important when thinking about urban NO2. So as we progress through this presentation today, I want us to keep these results in mind, especially as we think about what's causing our NO2 disparities and how we might address them. The way that we investigated NO2 disparities during the pandemic was by using satellite NO2 measurements and combining these information and these data with demographic information from the US Census Bureau. We're specifically using satellite measured NO2 from a satellite instrument called TROPOMI, which stands for the Tropospheric Monitoring Instrument. As you can see in this animation, as TROPOMI is orbiting the Earth, it's taking measurements of NO2 in the atmosphere, along with other key atmospheric constituents. And what we specifically do with these satellite data is we define two different periods. The first is a baseline period, which spans mid-March through mid-June of 2019. And the second is a lockdown period, which spans mid-March through mid-June of 2020. We then calculate census tract level average NO2 for these two different time periods for each of the 72,000 or so census tracts in the US. This next plot zooms into all urban census tracts in the US and looks at the demographics in census tracts that had the largest versus smallest drops in NO2 during the pandemic. I want us to focus on this bottom left subplot, which shows our racial background category from the Census Bureau. And if you look at the top bar in this subplot and compare and contrast it with the bottom bar in this subplot, you'll see that urban tracts with the largest drops or decreases in NO2 during the pandemic had about 30% fewer white residents compared with tracts that had the smallest drops. You'll notice that the percentage of residents who identify as Black in these two different categories is the same between largest and smallest drops. And what's really driving these changes is this other category. Now, of course, other isn't a race, and we are using it in this context to encompass many of the smaller categories that are defined as races from the US Census Bureau. Specifically, what's driving this difference in the other category is the number of people who identify as either being biracial or identify as having 3 or more races. If we look at the top right subplot, which corresponds to ethnicity, we also find that urban tracts with the largest NO2 drops have over 100% more Hispanic residents compared to tracts with the smallest drops. I think these results are really interesting, but what they don't quite contextualize is how these pandemic-related drops fit with the overall concept of NO2 disparities across different races, ethnicities, or socioeconomic statuses. So we'll take a look at that next. You'll see a couple of different conglomerations, as well as several different major cities in the US in this plot's vertical axis. And for each of these different locations or cities, you'll see 2 different bars. The solid color bar corresponds to our baseline pre-pandemic period, and then the dash bar corresponds to the lockdown period. And you can consult the legend in the right to see what the different colored scatter points correspond to. So the first take-home point from this slide is that least white communities in these different cities or areas consistently face higher levels of NO2 than the most white communities. The second really important take-home point, which maybe isn't quite as readily apparent, is that NO2 levels were higher in the least white communities of certain cities in the US during the pandemic than NO2 levels prior to the pandemic in most white census tracts. So what this means is even this global pandemic, with all of its very unprecedented and stark impacts on urban activity and mobility, could not undo these deeply entrenched, persistent NO2 disparities. The last part of this presentation is going to focus on what's driving these NO2 disparities. And in order to understand this, I want us to think back to the first slide I showed in this presentation, which emphasized the importance of on-road light-duty vehicle traffic in explaining NO2 in urban areas. Not only is traffic important to explain NO2 in urban areas, but also the placement of highways and roads and interstates is also really important when thinking about these NO2 disparities. We're next going to have a little history lesson, and unfortunately it's not the most positive history lesson, but rather speaks to some of the racist urban planning that has underpinned development in the US. The unfortunate truth is that the Eisenhower era interstate highway system often routed highways through minority and marginalized communities. And you can see a pretty powerful example of this in these two images from the Detroit Historical Society. The image on the left is taken from the Black Bottom neighborhood in 1959. This neighborhood had many, many thriving businesses, predominantly Black owned. And if you now look at the image on the right, which is taken just a couple years later from more or less the same vantage point, you can see all that beautiful housing stock and those thriving businesses have been bulldozed or demolished and now have been replaced by what, frankly, at least to me, looks kind of like a concrete hellscape. So this type of urban planning is not unique to just Detroit, and it occurred all over the US in cities. And if we take more of a systematic look at the placement of highways and interstates in relation with our pandemic-related NO2 drops and our demographic variables, what we find is that urban tracts that have the largest pandemic-related NO2 drops and the least white population have about 5 to 6 times more primary roads, which include highways and interstates nearby, than census tracts that had the smallest NO2 drops and the most white populations. You can see that in this figure. If you're looking at the left-hand side of the figure, what you're seeing are the number of highways and interstates that are located in communities that had the larger NO2 drops or lower income or lower educational attainment, et cetera. And you can contrast this higher number of highways and interstates on this left-hand side of the plot with the number over on the right-hand side of the plot, which is going to represent the number of highways and interstates in the more affluent, whiter parts of urban areas. I should also mention at this time that we've looked at the location of other sectors that are important for NO2 and NOx emissions, such as airports and ports and railroads. And we generally found that these other sectors were not preferentially skewed to be located in marginalized areas. So these results kind of single out traffic as being really important in thinking about the unequal and equitable distribution of NO2. So to wrap things up, what I've talked about today and what I've shown today is that marginalized communities in the US experienced temporary improvements in their NO2 levels during the pandemic. However, these communities continue to face higher levels of NO2 than communities that have a higher percentage of white residents, higher educational attainment and income. And thinking ahead to future policies that might address these disparities and their associated public health damages, what we've shown is that the 50% so drop in passenger vehicle emissions could not undo these disparities. And in many cities of the US, this 50% drop in passenger vehicle activity really didn't impact disparities all that much. So future policies are going to have to look into sectors that simultaneously are located in marginalized neighborhoods and also are important for overall NOx emissions in urban areas. And one candidate for a sector that meets those two criteria is looking at heavy duty vehicles and diesel trucking. So in keeping with the format of these talks, I'm going to spend the last minute or so talking about my career trajectory and research interests. So I have slowly but
a minute to go, Gage.
Okay, thank you. I've slowly but surely moved away from the quote unquote traditional atmospheric science and meteorology that I studied as an undergrad. And during grad school, I did a bit of work looking at how meteorology and atmospheric science impact air pollution. And during my postdoc at GW, you've heard what I've been doing and it's definitely more on the applied side of the air quality research spectrum. I should mention too that I'm on the job market, so as I look for positions at a college or university, I'm excited to hopefully continue this very societally relevant research on air quality, air pollution, and disparities. In terms of research interests, you heard about my interests on environmental justice and air pollution. I'm also interested in the impacts that transportation has on urban air quality and pollution. And I'm also very much interested in continuing some of the work I did during my PhD to try to understand the links and relationships among weather, climate, and air quality. So that's what I have for today, and I'm happy to take any questions that people have. Thanks.
Thanks so much, Gage, for an interesting presentation. We have some questions. Our first question is from Erica Garcia. I may have missed this, but were you comparing NO2 declines by % change or absolute change?
Good question. We did both. So in the bar graphs I showed, I can go back to that. We mainly looked at the absolute declines in NO2 and classified tracks as either having the largest absolute or smallest absolute drops in NO2. We've also looked at the percentage change as Erica, I think was the name, mentioned too. We get overall similar results to that. Of course, the magnitude's a little bit different.
Thanks. The next question comes from Alan Robinson. Very nice presentation and powerful results. It seems like the disparities were reduced by about half, while light-duty passenger cars contributed much less than half of NOx emissions. Is that correct? And what do you think the next most important category of sources to reduce?
Yeah, the precise magnitude at which disparities were reduced really depends on the city you're looking at. So on that bar kind of plot with the circles on, I only showed a few cities just not to clutter things up too much, but we've looked at a number of large urban areas in the US. Some of the cities have much larger than 50% drops. Some of them have much smaller. So you can kind of glean from this plot here. In terms of other sectors that are important, I'm not denying that light duty passenger vehicles aren't important. They're of course important for overall NO2 levels, but based on the fact that they declined by a pretty considerable amount and we really didn't eliminate or in many cities reduce disparities by all that much, means that some of the other on-road activities might be important as well. I should also mention too that although some of the other industries like ports and airports and factories are not preferentially located in marginalized neighborhoods on a national basis, these different industries could still be really relevant for disparities and environmental justice within individual communities.
Thanks so much. I'll turn it back to Anna Moon now.
Great. Thank you, Ellen, and thank you, Gage, for this presentation. Our final speaker today is Wiran Yushi, a PhD candidate in epidemiology at the School of Population and Public Health of the University of British Columbia in Canada. Her research focuses on linkages between the built environment and human health, with specific interest in transportation-related air pollution, the health impacts of air pollution and relationships between multiple exposures, for example, green space, mediated by urban form and population health. She has been involved in numerous exposure assessment and epidemiological studies with a focus on pregnant women, children, and the elderly. The title of her presentation is Neighborhood Environmental Exposures and the Incidence of Attention Deficit Hyperactivity Disorder, a population-based cohort study. Wiran, please go ahead.
Thank you for having me. So I'm going to present our project, which is the associations between multiple neighborhood environmental exposures and the incidence of attention deficit hyperactivity disorder. So attention deficit hyperactivity disorder is characterized by neurobehavioral symptoms, such as difficulties with attention, impulsive behaviors, and hyperactivity. So globally, ADHD is among the most. ADHD is among the most prevalent neurodevelopmental disorders, affecting approximately 5 to 10% of children. So over the life course, ADHD may be associated with impairments in social functioning, academic performance, and occupational stability, which have considerable impacts on individual well-being and healthcare, also the economy. So the full pathophysiology of ADHD is not clear. So previous research has suggested a genetic component, as well as environmental risk factors, such as exposure to green space and air pollution. The green space, such as urban parks and domestic gardens, may have protective effects on ADHD through mental restoration. And air pollution may be associated with increased risk of ADHD through neuroinflammation. Previous epidemiological studies linking environmental risk factors with ADHD often relied on cross-sectional study designs with small sample size, and the results have been inconsistent. Therefore, we aim to use a population-based cohort study to investigate the relationships between environmental risk factors and the incidence of ADHD. So this is the province of British Columbia in Canada. Our study area is Metro Vancouver. Metro Vancouver occupies the southwest corner of mainland British Columbia. There are about 2.5 million people living in the Metro Vancouver area. Our cohort is population-based and assembled from administrative data sets. There were approximately 37,000 births included in our cohort between 2000 and 2001. Here is our study timeline. So the baseline started from birth between 2000 and 2001. Exposure period was from birth until the age of 3. Our primary exposure is green space, which was measured by satellite-derived vegetation percentage with a buffer size of 250 meters. So the majority of epidemiological studies have relied on NDVI. For exposure assessment of green space, NDVI is the normalized difference vegetation index. So although NDVI provides an objective measure of healthy vegetation, but there is an issue of mixed pixels, that different materials jointly occupy a single pixel. Mixed pixels lead to difficulty in capturing small patches of green space, for example, pocket parks or domestic gardens. On the other hand, the vegetation percentage was derived from a linear unmixing process, which identifies regionally calibrated pure pixels and provides a more suitable estimate of vegetation coverage compared to NDVI. Moreover, vegetation percentage ranged from 0 to 100% and provides a higher range of variability and is more easily interpreted compared to NDVI, which is a ratio ranging from -1 to +1. Other exposures were PM2.5, nitrogen dioxide, and noise, which were estimated from land use regressions and the deterministic models respectively. Our follow-up period started from age of 3 until 2010. Because children can be diagnosed with ADHD as young as 3 years old. Incident ADHD cases were identified based on a combination of criteria, including physician visits, hospital diagnosis, and drug prescription. We adjusted a total of 14 covariates in the model. These covariates were selected based on previous literature. In total, we identified 1,217 ADHD incident cases. Green space has protective effects on ADHD. For every 12% increase in vegetation, there was a 10% reduction in the risk of ADHD among children. And air pollution was associated with increased risk of ADHD. For every 2 micrograms per cubic meter increase in PM2.5, there was a 11% increase in the risk of ADHD. So to understand the potential joint effects between green space and PM2.5, we stratified green space and PM2.5 into 5 different quantiles. So these are the results. In brief, there was a 50% decrease in the hazard ratio for ADHD in areas with the lowest PM2.5 and highest green space exposure, compared to a 62% increase in hazard ratio in areas with the highest PM2.5 and the lowest green space exposure. There were some joint effects between green space and PM2.5 on ADHD, which suggested that there were environmental related inequalities in neighborhood in which children living in neighborhood with highest PM2.5 and the lowest green space were at much greater risk of ADHD. Our results further suggest that green space initiatives in urban planning may reduce negative effects of harmful exposures on ADHD incidence among children. So next, I would like to briefly talk about my background and the research interests. So during my bachelor program, my first research project was to identify sources of air pollutants and derive multiple predictors of air pollution from remote sensing data. And for my master thesis, I used statistical models and machine learning techniques to build predictive models in order to estimate indoor air quality among pregnant women. Meanwhile, at BC Center for Disease Control, I was involved in several environmental health projects. For example, we created the heat index to quantify neighborhood risk during extreme hot weather. My PhD thesis was about associations between air pollution, green space, and dementia in Canada. I also collaborated with researchers from the Department of Forestry to study health effects of green space. I am interested in applied public health research with interests in air pollution and built-in environment in support of evidence-based environmental health policy. And I aim to become an environmental epidemiologist. Thank you.
Thank you, Viran, for this very interesting presentation and giving us your background. We unfortunately lost the questions to our previous speaker, and I am not seeing any questions for you at the moment. Can you tell me a little bit more about how you became interested in the neurological outcomes, the ADHD and also dementia?
I think I was interested in air pollution, and air pollution has various adverse effects on multiple organs. And the previous evidence and studies were well established on the associations between air pollution and lung cancer, cardiovascular disease, and respiratory disease. And just recently, air pollution was identified as a risk factor of dementia based on the most recent Lancet report. And so therefore, we believe that the evidence was extensive on the adverse health effects of air pollution on heart and the lungs, but now we shift our focus to brain, and because air pollution can trigger inflammation in our body, which will impact multiple organs. So we believe that brain is also impacted by air pollution. So that's why we are sort of conducting epidemiological research to see if air pollution has any adverse health effects on dementia, like incidence or mortality.
Mm-hmm. Thank you. There's a question from Chad Bailey. Fascinating study. Could historical lead deposition be a confounder in the LUR and green space exposures?
So that's a good question. We did not consider this lead deposition in our model as a confounder. So we did not, at the moment that we completed the study, we did not find lead deposition in the literature to be a sort of risk factor for the incidence of ADHD. So therefore, I don't have a clear answer for you.
Okay. I'll go to a question from Alison Patton about the pixels. Can you explain again what mixed pixels are and why NDVI has mixed pixels and your method does not?
So the mixed pixels, as I said, the issue of mixed pixels is that, you know, within one single pixel, there are different types of materials. They occupy the single pixel, and the NDVI does not distinguish them. But the vegetation percentage, which was derived from a linear unmixing process. So this process can actually sort of extract the exact pixel related to green space or vegetation from the single pixel. So that's why it's more accurate and precise. And some researchers believe that, you know, vegetation percentage derived from this unmixing process is actually a novel measure of green space compared to NDVI.
Okay. Thank you. A question from Carol Henry. Have you looked at other health effects that are mitigated or influenced by green space?
That's a good question. Yes, but not in this cohort. So my PhD dissertation was about air pollution, green space, and dementia. And the results are like the preliminary results are like green space also has protective effects on the incidence and the mortality of dementia. So if I remember correctly, so green space, so we used NDVI, but so basically for 0.1 unit increase in NDVI is associated with, I think, 3% decrease in dementia mortality.
Mm-hmm. Thank you. I think that answered Martha's question as well, unless I'm wrong. I'll go to Jeff Brooks' questions. Based upon what you have found, do you have thoughts on practical interventions that could reduce such outcomes?
So I think one of the possible interventions is to be more careful about the urban planning. So recently, I think because of urbanization, so there are many developers are building high-rise around like SkyTrain or subway, and in which they may have a small garden, but it may not be adequate. So I would say, you know, one of the interventions that I can think about is to add more green space into urban planning when approving new buildings, like such as high-rise or low-rise around subway or SkyTrain stations.
Mm-hmm. Thank you. Question from Martha Ondras. Do you know how sensitive the administrative database is in diagnosing incident ADHD? Could there be different rates of diagnosis between populations that are in highly polluted versus lowly polluted areas?
And that's a good question. It's actually one of our limitations in this study, because we acknowledge that based on the criteria using the administrative data sets, we definitely underestimate the number of incident cases of ADHD in our study. Because despite we have a combination of criteria, physician visits, hospital diagnosis, and the drug prescriptions, but, you know, people may not go to clinics, or children, they just stay at home for treatment, or there are some missing values. So I believe that there are some differences, and our numbers are underestimated.
Mm-hmm. Thank you. I'll do a last question from Eloise Marez. Have you shared your results with city planners and other relevant decision makers? If so, what was their response?
Oh, thank you. So that's a good suggestion, and we haven't done so. The work was submitted and is currently under review. So that's why it's not published yet, so we have not shared it with other urban planning or decision makers. Yeah.
Okay. Thank you. I'll hand it back to Ellen for a minute so we can reopen the Q&A for our previous speaker that was cut short.
Yes, and I apologize. You know, no matter how much you practice for things not going wrong, obviously things sometimes go wrong. So unfortunately, all the questions disappeared in front of my eyes at the moment, but there are some good questions that didn't get addressed, and I'd like to go ahead and ask Gage to come back and answer a few of these. So I apologize to Gage. The question comes from Stephen Chilrud. He says, are you going to extend this analysis to take into account meteorology impacts on these estimates in each city? The recent publication on Beijing impacts give a nice example of the importance of doing so, and it is in your toolkit.
Great question. We've done some sensitivity analysis looking at if the 3-month period that we use for a baseline and lockdown periods is sufficient enough to smooth over different weather patterns that might bias results. And what we found is that NO2 disparities calculated with that 3-month period that we've been using closely resemble disparities calculated using longer time periods. So although we haven't applied any type of machine learning correction or other type of like analog correction to adjust for meteorology, we're pretty confident that looking at these longer time averages doesn't lead to more biased results from the different weather patterns.
Thanks. The next question comes from Eloise Maras. It says, really important work, Gage. Did you look at aerosol optical depth changes as a way to assess the impact of lockdown measures on primary PM 2.5 from roads?
No, we haven't. That's a really good suggestion. Our analysis so far has just been looking at satellite-derived NO2 as well as some surface-level NO2 metrics as well. But looking at AOD or other pollutants would definitely be good moving ahead.
And then two sort of related questions that I think are important. One from Allison Patton that says, thank you, Gage, for this presentation, and the figures communicate a clear story. Can your results be applied prospectively to develop more equitable air quality management policies for the future?
I guess I'm biased, but I think the answer is yes. This goes back to what I was talking about the importance of on-road heavy-duty traffic in explaining these disparities. I think moving ahead and thinking about policies addressing this on-road heavy-duty traffic might be a good place to start. I think that over the last decade or so, passenger automobiles have gotten cleaner and cleaner, and some of the heavy-duty diesel trucking still remained a pretty dirty source of NO2. So looking at these sources, I think, would be pretty important.
Thanks. And just one last question then from Inyang Uwak. Great presentation, Gage. Any recommendations to improve or reduce the NO2 disparities in the US while waiting for policies?
Ooh, that's a good one. I think, I'll punt back to the kind of common thing we hear about, reduce, reuse, recycle. I think the more that we maybe rely on public transit or use more active forms of transportation to get from point A to point B can't hurt overall NO2 levels. I do feel that policies from the top down that address some of these other sources of NO2 beyond passenger vehicles are urgently needed, but that's not to say that making choices about lifestyle and how we commute won't affect overall NO2 levels.
Thank you. And thank you again for your patience and a little technical difficulties. I'll turn it back over to Joanna for some closing remarks.
Thanks, Ellen. I'd like to thank everyone for attending our webinar today and for all of your excellent questions. Just a reminder that the recordings of all the webinars as well as the slides will be available on our website in the coming weeks. And we'd love it if you could take a few minutes tomorrow to complete the post-webinar survey that you'll receive in your inbox to help us improve our webinars for the future. We hope you can join us for our final webinar of the 2021 Annual Conference webinar series, which will take place next week on May 25th. And finally, we hope to see you all at our in-person conference in Washington, D.C. next year from April 24th to 26th. Thank you again on behalf of all of us at HEI and enjoy the rest of your day.